I'm Chuck Cywin. I'm Director of Small Molecule Neurotherapeutics Development at NINDS, and have been responsible for the Blueprint Neurotherapeutics program since 2017. I've been with it since almost its inception in 2012. This program this course developed also out of the Blueprint for Neuroscience Research, and is designed to give you sort of the foundation of what you need to learn about to come to the programs. It's also been expanded. Now you obviously are seeing biologics, and I'll talk today about both small molecules and biologics funding opportunities. Oops, where's my thing? So, first disclaimer, all opinions are my own, not those of the government. I am opinionated, so you'll get some things that don't always there. I have no conflicts of interest in terms of financial things to report. I am going to talk today about the space over here from the bench to bedside. You'll hear translational research discussed in the whole paradigm from both bench to bedside to community. This is focusing in on later stage clinical trial work as well as comparative work, and our audience here today is really more in this earlier stage, and that's where we'll all focus on. NIH spends a lot of money in neuroscience research, and it spends it across multiple different institutes and centers at NIH. And this gives you a graphic from, I believe, 2020's budget, and you can see there's some big players. Aging is in there, neuroscience, mental health, the Eye Institute, drug abuse are all major players in this space. And the ones that are highlighted in blue on this slide are part of the Blueprint programs, and for which is funding this course, amongst other things. So what did you learn in this course? You hopefully learned a lot of things beyond what's on this slide, but there are certainly multiple modalities that are neurotherapeutics, and small molecules and biologics are the focus for today, but there's also things like devices and combination therapies of those that go in there. And starting compounds are obviously rarely drugs, right? We always need to optimize things for ADME, for potency, for brain permeability, for toxicology, for exposures, and you heard all about that. You also need to be able to have intellectual property, which you heard about today on the things. So you have to think about what the regulatory requirements are to get your particular project to man. And it's not always the same, right? For the IND pathway may be the pathway for some projects, but not always. And so you have to think about what that pathway is going to look like, because that's going to dictate where you're going to look for funding to do it. So take-home message, drug discovery takes time, money, and expertise. And NIH has the ability to help you provide all three of these things to you. And I'm going to tell you a bit today about some of those programs. It all starts with you. You guys bring us new targets. You bring us new ideas for small molecule pharmacology, biologics, and devices with good pharmacology. And as I said, I'm going to hopefully guide you through this all. I'm going to talk in terms of my outline. I'm going to talk about the Blueprint Neurotherapeutics specifically, because it's what I'm most familiar with internally, and give you an example of how the project helped an individual project move forward, and also increase your awareness in terms of opportunities to move your projects towards the clinics for other modalities like biologics and devices. I will give you sort of an overview of the NIH organization, some things to think about, and then in the backup slides, which you'll have some helpful hints to be successful in there. I don't think we'll have time to get to all of that today. So the Blueprint for Neurotherapeutics Network, also known as BPN, is the program that I joined NIH for. I came from industry. Before I came to NIH, I worked as a medicinal chemist for 17 years at Behring Ingelheim, and then I moved up in the ranks there. I was director of medicinal chemistry, and I got an opportunity to come to the NIH after I left Behringer to start in this program where the idea was we want to bring industry expertise to our applicants. And how are we going to do that? We're going to set up a program for which we have hired expert consultants to help the team members. We have staff that understand drug discovery and development, and we have funding to do that. And put all three together and be able to hopefully move projects to the point where we get them in the clinic and get them licensed. The end goal, and what was really unique about the program at the time, is this is an extramural. We're doing it with people outside of NIH scientists, and we're trying to maintain the intellectual property so that you own it. It's not encumbered, and the end goal there is that you'll be able to get it licensed. And again, get to people with deeper pockets, move it on to the clinic, and hopefully into helping man. So we want to decrease the risk, and we want to obviously always have a handoff. There are a number of institutes and centers that are signed on to these programs. These are the ones that are signed on for the small molecule space, and you can see we cover 9 different institutes and centers here, and the contacts for each one is listed in your slide deck, which you'll have available to you. So have we been successful? And I decided I'd add this slide in this year to say, yes, we have been successful. To date, we've run 38 projects covering 8 different institutes and centers. We have 7 of those projects that moved on to the clinic, including first-in-human trials. 2 projects are now in phase 3, and hopefully we'll see registration for them in the not-too-distant future. One is for Fragile X and cognitive impairment in Alzheimer's disease. That project was started at a small business and was licensed by Shionogi initially, and then the whole company was bought by Shionogi, and are doing the clinical trials now. And then we had a project in Stargardt's and age-related macular degeneration. I'll talk more about this project in a minute. That is with Belite Bio. And we also have projects with major depressive disorder that actually spun out of a project that we have that was licensed and repurposed for that indication after us. And then we have things for hearing loss, which are now moving towards continuing their phase 2 work. They had some setbacks, but they're going forward, and projects in phase 1. We have additional INDs coming this year, and we've also assisted projects ad hoc by teams that have come in and just used our contract resources to move the projects into the clinic. It's also been very successful in terms of getting licensing. Many projects have been licensed to date, and the value of those license deals is probably close to $2 billion in potential investments. So how do we do this? As I said, we put together teams, and they're custom tailored to your particular project, and it includes our lead development teams have the principal investigator sitting on them. They have consultants that we assign, and the consultants are custom tailored to your particular project based on where you're coming in. So if you're in a small molecule space, it would be like a medicinal chemist. If you're doing optimization, a DMPK type person, maybe an assay development person. But as you progress down, you're going to need toxicology expertise. You're going to need some regulatory help. And you need process and formulation work, and so you'll get people that come in there. And then you have NIH staff here helping to facilitate this, and these are done through cooperative agreement mechanisms. We can plug and play. If you have these resources yourself, we can do some or all of the contract work, and the contract is handled separate from the grants. And that helps in terms of if you spin out companies and things. Yes?
Forgive me, I'm not super familiar with this. So if I have a grant, I contact my program officer?
No, this is a cooperative agreement. This is a grant mechanism itself.
Okay, it's its own thing.
And it has both grants and access to consultants and access to the CROs is all part of the program. It is theoretically possible to access the contracts ad hoc for ongoing work, but the institute and center has to want to do that. And it becomes sort of a supplement if you have an existing grant to that. You would have to talk to them about it. It's not something we do typically, but on rare cases it has happened. Again, we're trying to retain intellectual property. What I should point out here is, it is a phased cooperative agreement mechanism, it's either a UG3, UH3 mechanism, or if you're coming under the SBIR, it's an SBIR fast track, phase 1, followed by phase 2. There are different budgets associated with them, different rules associated with them, but the program itself runs identically. And you just have to be sure which of the two programs you need to go into based on what you are. If you're an SBIR eligible organization, you come into the SBIR side of it. If you're a small business who's not SBIR eligible, you can still go to the UG3 and UH3. If you're using foreign components, you're going to have to go to UG3, UH3, and type of things. There is a little note up here, and I highlight it here, that not all ICs accept development projects. For instance, mental health and drug abuse do not accept development projects. They have their own FOAs for those type of things, and they prefer you come only to discovery projects into this. Likewise, there can be other restrictions, like NIDA has restrictions. You utilize the contracts to the maximum extent possible as you're doing it. So do talk to us about these if you want to come in. It is important to know that these only have receipt dates twice a year. The next receipt date's in August. As I mentioned, it's a phase mechanism. Everyone comes in the preparatory phase. Everyone, so even if you're starting at assay and I have a hit, you have that early phase, or have a development compound, there's going to be work to finish out your milestones to move things forward and figure out. Everything is done on a staged mechanism so that we're investing money based on making progress. The phase 2, again, we support up to a couple of years of medicinal chemistry in that phase, so you can get a maximum of like 3 years of medicinal chemistry support in doing this type of work. I just mentioned they come through both. In all cases, though, we are trying to get to an IND. We are trying to get to that first in human trials. We have a clinical trials contract. We can run healthy human subject clinical trial, first in human studies through that contract. And so in all projects, we want to get to that. That is a common inflection point for investment, is to be able to get to that first in human, get human PK on it. It does not allow for simultaneous backup programs and front runner programs. So we're going to push the project forward as fast as we can to the clinic and move things forward. And the goal in all cases is that we want to have a balanced program. So I did mention milestones. Oops. And this sort of lays out. And on our website, you will see there is, in the frequently asked questions, a thing about milestones. It has what you see on this slide, as well as a breakdown individually, what goes in each one of those sub-bullets there and what we're looking for. So you can get an idea of how a project would be laid out in small molecule space. Similarly, I think it's available for biologics. In the interest of time, I won't go through these testimonials. They're up on the slide deck if you want to read or hear what some of our investigators have said about being in the program. They're available to you on these links, and I'll do that. What I do want to do is give you an idea of a success story and how we did it. This is Costa Petrukhin. It came as an academic project coming from Columbia University. Costa came into our program with the idea of doing AMD-related work on RBP4 blocking its interaction with TTR. There was work from Amgen in the literature on this for diabetes. The proof of concept was there that it worked and that it could be done, but it hadn't been done for the eye indication. It had been dropped because it didn't work, I think, in phase 2 from Amgen, and there really wasn't a lot of work done beyond what was already published there. What this project became was take a literature compound and move it forward for a new indication, which means you have to develop new intellectual property, you have to get your own active species and make that things work. How it works is simple. It binds to RBP4. It breaks up the complex of RBP4, retinol, and TTR. That allows the retinol to get degraded. Otherwise, it goes through the visual cycle and produces cytotoxic cytotoxic retinoids that eventually lead to dry AMD. So as I said, they came in with a compound that's in the literature from Amgen, and we looked to conformationally constrained analogs of these and other stuff. But our key issue that we identified from Amgen in their literature that was out there is they had some metabolic stability issues associated with their molecule, and we had an intellectual property issue. The compound belonged to Amgen. And so early chemistry focused on those IP things. And how do we do it? So the chemists that worked with us at Curia came up with some BIS or some fused ring systems that were putting the key pieces in the right places, and we're able to develop a new scaffold. This new scaffold demonstrated good activity and patentability and freedom to operate, and that was our first step moving forward and showing a proof of concept. That was done in the preparatory phase, or would be the preparatory phase today. And in fact, it does improve metabolic stability of the molecule. You can see in humans, we went from 3% remaining for the Amgen compound to 100% remaining in the microsomal stability assays here. So we thought we were on the right track. However, as is always the case in drug discovery, you fix one thing and something else pops up. And this was here. We had CYP inhibition, which we did not want. So we continued to do optimization of this molecule. We did show that it had good exposure in rats, and that we had good bioavailability, and all that was moving things on, but we did need to fix the molecule. And this is where developing a solid screening funnel is essential to moving a project forward. And we worked with this team to do that. The lab, the home lab of the academic group is in the greenish blue here, and they're doing those assays. AMRI was doing all of the synthesis, the in vitro ADME work on the project, and then we had multiple different contractors doing PK and other work for us to move molecules forward. That's how we brought the team together with the consultants, and for assay expertise helped improve their assays, make them higher throughput, and the medicinal chemistry and PK all came together. So moving forward, we went from this group where we had the fuse system. We were eventually able to replace it with different changes here to get to an improved compound, and eventually showed that across the board we had improved potency and CYP profile on the molecule. We took this molecule further down the pike. It's published in JMed Chem back in 2015, and showed in proof of concept in monkeys that, in fact, it does reduce the RBP4 levels significantly and for a long duration after a single dose. And in fact, it turned out too long a duration in this particular molecule, and that it caused some issues, and that we decided to look for a compound that had a better balance of the profile. So we continued to do SAR. We eventually got to this molecule, 14967, also known as LBS-008, or I won't even butcher that name, sorry, that is now in the clinical candidate for this molecule and is now in phase 3. So the outcomes that we showed here and worked with the teams on is they were funded by us. We made 500 molecules for them to optimize. They produced, I think, 6 or so patents in different scaffolds for the team. The expertise on this helped move the project forward, and a deal was announced in 2017 with Lin Bioscience, whose parent company is Belite, who you saw referenced earlier. Lin has partnered and moved this project forward into the clinic. They worked with us all the way through phase 1 as part of the LDT, and then took over and continued the project after we were done. And I just have some headlines of it, and we just watched it move forward. It had, they were able to get additional funding in millions of dollars to continue the phase 3 funding. It's in phase 3 now, and we hope to see it continue on. This is just a acknowledgement of all the people that worked on it. It was not just one single group. It was Columbia Group, it was the consultants group, it was the NIH staff that all moved this project forward to do this. So this is an example of how NIH can help a project move forward. We have programs across the whole spectrum of drug discovery and development. Not all of them are cooperative agreements like this with Blueprint, but many are. And so I'll talk a bit more about some of the ones in red here, which are in my division, Division of Translational Research. But keep in mind, there are other projects that help with moving projects forward in the later stages of clinical development. So I should, and all you folks interested in biologics space, say this program for small molecules spawned programs for biologics as well that just launched actually in 2021. So the same program is being set up now for biologics. We have contracts in place to do the same work. They're utilizing either the same clinical contracts that we have already in existence, but they've also added contracts that have flexibility to support biologics in all modalities of biologics available. Their website is down here. Chris Boshoff is the program director in charge of it right now, and we're moving that forward. The IGNITE program is a program that we have at NINDS in order to get you the preliminary data that you need to enter these programs. And there are 3 different funding opportunities under the IGNITE program to help you this. One is an assay development. So develop an assay, run a screen. That gets you the chemical starting point or to move things forward. Or you can have neurotherapeutic agent characterization in vivo studies. It's about optimizing a molecule enough to have a tool compound and running the efficacy study in order to show that you have good preliminary efficacy to support your particular project going to a more advanced program. And then if you need animal model development, develop that and validate animal model is available through a third funding opportunity. These are all phase mechanisms. They are not cooperative agreements. They're R's, not U's. So there are R61, R33. They have limitations on the budget. Becky Roof is the program director for this program, and they have multiple receipt dates per year. And it's a great program to get you the data you need to have a better chance for getting good score in the review for the Blueprint programs, amongst others. Not to be left out, the devices, which I know are not the focus here today, but there are combination things that come into play here, and sometimes the device program is a place to consider, and we have certainly put in applications for nanoparticles and other things into device space in the past, as well as handling them through the SBIR program. I've talked to you about NINDS programs so far, and the ones that really support the neurotherapeutics development, but we're not the only ones that do that. As we say, NIA has a whole suite of programs directed towards Alzheimer's research. They also participate in the Blueprint Neurotherapeutics networks, and they have their own in there. And so it is prudent to talk to NIA if you're in this space about what they think is the best program for your program. I always tell people who are trying to figure out whether they should come to Blueprint Neurotherapeutics or go somewhere else, really ask yourself, do you need the resources that we have? Do you need the expertise that we are going to provide? Or do you have that already worked out in your group? If you already have that worked out, then you may be able to do it faster doing it outside of the Blueprint. If you need that expertise, we're a great resource to do it within it. On top of that, there are other trans-NIH programs that are in neuroscience space that are outside of Blueprint. Things like the NIH HEAL initiative, which is directed towards opioid use disorder and pain therapy. There's a great program that was actually spun out. It's based on the Blueprint Neurotherapeutics Network called the Pain Therapeutic Development Program. I'll talk about that in just a moment briefly, but it's part of a broader program. It starts with planning for therapeutic development, doing the initial therapeutic development, things like getting the mechanisms, the assays, the models in place, as well as the optimization stage, which is the Pain Therapeutic Development Program, which is really essentially BPN plus a biomarker component for pain and opioid use disorder. This is actually, sorry, I shouldn't say it. This is just for pain, opioid use disorder. There are similar programs in NIDA for opioid use disorder as well, and I'll perhaps touch on those in a bit. I guess the important thing to note is they have different ICs signed on to this, but do not, if you're in pain space, do not be concerned if your IC is not listed. If you're in pain space, you're in the purview of HEAL. And NINDS will take care of taking the lead in terms of science officers on a project, for instance, for chemotherapy-induced pain. You don't see NCI listed here, but we have plenty of projects in chemotherapy-induced pain. So that's fine. The R61 goals for this program are, multidisciplinary, set up a multidisciplinary team, show me the biologic rationale and feasibility, show me the clinical benefits of the potential pain therapy, and map out the plan. So it's sort of that preliminary project team. The U19 is really is a big, large, multi-component program where you're bringing in interdisciplinary team together, and it's a cooperative agreement mechanism, and again, milestone driven, but it's like Ignite in that regard, but bigger. You're putting together a whole team, and you're trying to get the project to the point where you're handing it off to the Pain Therapeutic Development Program. Attrition is expected in these programs, and you will see projects stop based on milestones. The Pain Therapeutic Development Program I just wanted to mention is not just small molecules, it's small molecules and biologics, and therefore has the full access to both sets of contracts that I mentioned in Blueprint. So you have not only all of the curia and toxicology folks in here, but you also have the contractors for biologics like Leidos, Battelle, and ABL that can help with discovery and development in biologic space. Everything, again, is leading to clinical contracts and clinical studies. We want our projects to move into the clinic. The handoff for this program is hopefully going to be EpicNet to take your projects into phase 2. My colleagues wanted to make sure that I mentioned there is a webinar coming up on April 12th at 2 p.m. You're welcome to sign up for that and learn more about the HEAL program. So I'm going to shift gears. I talked about the programs. I want to talk to you a bit about the NIH and how things work. And you guys are probably pretty well familiar with it, but there are 27 institutes and 24 provide funding. Each of them have different missions, different funding priorities, different budgets, different grant types. They're just different at times, and you have to be aware of this. And I heard the questions asked, how do I know where to go first? Where do I go? You go where your science tells you to go. Where do you have the best preliminary data to support a grant application? There's almost all of these projects will cross multiple different disease areas, and we talked about that target product profile up front. What is the data you think you have in hand that supports the project? That's where you should go first. That determines what institute you need to go to. Because as much as I'd love to say that we all work together and all these institutes function seamlessly together, when it comes time to trying to get them to co-fund individual things, the more institutes you bring into the co-funding discussion, the more complicated it is. So if you can get it to say, I have really good scientific data for Alzheimer's disease or Parkinson's disease or OUD, all right, it's easier to move a project in that individual IC and get support than it is for me to bring all those ICs together to get, one person says no, then what do I do? So it's better to just come in with a focused project. I know, and everyone knows in drug discovery, at some point, whoever's commercializing this is going to add indications because they want to market it to whoever they can. And we know that too. Everyone understands that. But it's where's my first set of patients going to be? So I always ask that when you planning, we talked about planning with the end in mind and with the clinic in mind. Where's my first clinical trial going to be and what patient population is going to be? That'll help dictate where you come to for funding. So I just talked about all that here in the first part, but also think about funding priorities, right? Things like the Brain Initiative by Alzheimer's disease and related dementias, like HEAL, they have their own separate pool of money and are going to fund things potentially with a more generous pay line and then the rest of NIH at times. Not always the case, but there are times that's true. So talk to your program directors. If you fall in those things in that area, you may want to be searching out those particular funding opportunities specifically first. As I mentioned, know what your target product profile is. The next slide is going to look fairly familiar because it is a target product profile, and we just talked about that before, so I won't belabor this, but other than say I do have this up on my website. I'm having trouble finding it on the FDA website. They've taken it down. So if you can't find it in the FDA website, you can find it on our website. It still exists. Where else can you find information, right? You can find information around here at NIH in a number of different places. We want to set up you for success, and so there's a lot of great information available to you 24/7 at grants.gov and other places. And one of the key places where you need to know where to go from the get-go, right? So before an application is been submitted, you can talk to program staff like me, and we can help you. We can help guide you. But once you submit the application, it's in the scientific review officer purview. I can no longer discuss that application with you while it's being under review, right? I can answer general questions, but I'm not going to be able to tell the scientific review officer what you're asking me to change, right? If you have a problem with something after you review or after you submit it, talk to your scientific review officer. Once it's reviewed and the summary statement's out again, you're back to me as a program officer, and I can go over what I saw in the summary statement and potentially, from my experience, say where you think you need to go to next or what the likelihood of funding is and what next steps might be. We do need to know, a collaborative team, as we talked about this, and you need to have experts in all these different things, and you may or may not be utilizing the Blueprint Air Therapeutics Network. So how are you going to find people that you don't know? Well, obviously, you're going to use your own networks as much as possible. But again, the NIH does have tools, and a lot of people don't ever utilize them. Things like NIH Reporter is a great tool, and it serves that function by, if you just do a straight-up search and look for things like neurotherapeutics, you'll get a number of different things. But if you click the View button on the grant that looks of interest to you, you drill down on those things, and it gives you additional information. Here's the name. You can click on the name of that. You can get into it. You can hit View. You can get into this next step. And what you get from this is project details. You can get project details, publications, all these different things on the side. If you click the Details tab, you can really find out the contact, where they're at, what their email address is. You can find out who the program officer that has this grant in their queue is right now. So that tells you who at NIH you need to talk to. And I know this is a hard thing for a lot of people to find. It's like, how do I find out what the right program officer for me to contact is? This is a great way to do it, right? You can drill down, and if they're not the right one, at least you're in the right, church, and if not the right pew, per se, all right? And they'll be able to figure out where you need to go. I have plenty of time. I can go through additional questions, but I'm going to stop right here for questions. I have a whole bunch of slides on sort of things to do and things not to do. But I'm going to stop right now for questions.
So can you expand on the HAIL program? It's a U or R? I mean,
It's a U grant.
it's a U. So how we can start? Like if we have, let's say we have a lead compound, where we can go from there? I mean,
Okay. So the Pain Therapeutic Development Program is RFA NS 21-010, and it is a UG3 UH3 mechanism. It has a 2-year UG3 in there in the preparatory phase, followed by a maximum of 3 years in the UH3. Just like before, you can enter the discovery phase all the way through to a development phase compound. I always caution people when they say, I have my clinical candidate, that I start to pressure test that question. Do you really have a clinical candidate at this point? Do you actually know you have one? Have you run dose range tox finding studies? Have you had PK at multiple doses? Do you know whether the PK as you dose up is linear? Do you actually understand all of that? Or do you have additional work that needs to be done? And then what happens if it's not? Are you going to want to come back and do additional optimization? So think about structuring your project. If you really don't know the answers to those questions, to structure your project that you're really in the optimization phase so that we can do the initial analysis to figure out whether we actually have a drug candidate while still continuing to optimize for the things we know might be a problem. And then stop that activity if, in fact, this is the clinical candidate. We're going to move forward. The other, like I say, the key thing about the HEAL program is they will look for the component of the biomarker, and they will fund you to develop. It's really a pharmacodynamic biomarker that they're looking for. Can you develop that for your particular project? So they move into the clinic. You heard it touched on today. Many companies in neuroscience space will not invest without some sort of biomarker. And not all companies are that way, but there are several that are in major pharma that will not invest in neuroscience without a biomarker to read out.
So quick question. So besides the requirement for the working with NIDS stuff, what's the difference in terms of SBIR phase 1 between the omnibus SBIR phase 1 and the U44 SBIR?
Well, the omnibus is an R grant, so that's just giving you money, and you're limited to the SBIR limits that you saw there for both phase 1 and phase 2. You have no access to consultants, no access to the contractors, which are funded separate from the SBIR. So the actual value of those projects can be up, we could potentially be putting up to $12 million into some of these things to get all the way through into the phase 1 clinical testing. You would not be able to do that within a simple omnibus in one. You'd have to do it in multiple different grant applications as you go through the process. Additionally, you can have troubles in SBIR space when doing drug development if what you need to do is considered R&D and you're not keeping it in your small business. Like if you have labs and you're doing a small business, no problem. But the rules are you have to keep 2/3 of the money in phase 1 in your company and greater than 50% in phase 2, unless it's a fee-for-service thing, which comes out of it. Many of these activities are not considered fee-for-service, and you cannot possibly meet the SBIR rules if you don't have your own labs in those situations. So virtual pharma companies that are small businesses prefer many times to come to Blueprint or one of these other cooperative agreement mechanisms where we're funding it sort of outside the group. Also, if they're not qualified for SBIR eligibility for those things, like you can't meet those requirements, you can come to the UG3, UH3 mechanism and fund it under that mechanism instead.
Hi, I know you are doing this small molecule, but for these folks who want to using the similar mechanism for like ASO, gene therapy, AAV, can you comment on this, like how people can seek NIH support from
Yeah,
this area?
like I said,
Thank
we
you.
set up the program for the Blueprint Neurotherapeutics Network for biologics with that in mind. So ASOs, cell therapy, gene therapy, antibodies, nanobodies, all of that falls under that particular program. And as you saw from the contracts, they'll run a little bit different because there's so many different modalities that I can't have a single contractor that I could say, like Curia does medicinal chemistry, and everyone coming in I can give to Curia. I can't do that with all the different modalities in biologic space. So we contracted with Leidos, Patel, and APL to be able to have that flexibility. So we can put out a statement of work associated with your particular project, let those three vendors figure out. And if they need to subcontract, they can subcontract to do that work.
Thank you. It's very informative. So I'm wondering, is it possible for the teams to hold a R01 discovery, but at the same time doing the blueprint for the development at this, in parallel, or kind of stack together?
To do development and discovery simultaneously?
Yeah.
No, you can't do that. Well, you can't do that within these cooperative units I talked about, Blueprint or the Pain Therapeutics Development Program. It's one or the other, because we view them as two separate programs. And because they're so costly, trying to do one, unless the other one is dined off, we really don't want to do them simultaneously. That's not to say you couldn't request funding for both. You can. You just have to separate them out to two separate projects.
Thank you.
Okay, any other questions for Dr. Cywin? Okay, well, before we break, I just wanted to recognize Dr. Cywin as our program officer for this course, and we're really grateful to have the collaboration with him, and thank you very much, Chuck, for all the effort that you put into making this course
You're
a
welcome.
reality.
I've been your program officer for this course up until this latest iteration of it. Now it's Arisa, who's not here today, who's worked with me as scientific project manager for the last probably 9 years, it seems, and is taking over that role. I took this over from Amir. I think took it over from Rebecca. We've all been associated with Blueprint Neurotherapeutics over the years, and we pass this along. But I am grateful for the opportunity to talk to you all about these opportunities. And it's just the opening dialogue. Feel free to reach out to myself or anybody you saw listed on here about the programs. We're always happy to set up calls. Don't be surprised if you respond to me that the next email you get is talk to Rakonda, because that's what happens. She manages my schedule and tells me where to go and where to be, including here. So I'm here today solely because Rakonda got me here today.
Great, okay, well, thanks so much.
Thanks.
Thanks.